Dear students, today we will discuss about the chromosomal aberration and the first part of that chromosomal aberration, that is structural changes. This topic is offered under the fundamental of genetics offered in second semester of BSc AG degree program. So today we will discuss all about this structural chromosomal aberration. So before that we must know about the chromosome. It is a teeny thread-like structure, and almost all living cells have this type of structures, and which carries the genetic information in the form of genes. And this chromosome is made up of histones and DNA. The chromosome, first described by the Strasburger in the year 1875, they don't use the term, the word chromosome firstly, but later on, Waldeyer in the year 1888, they coined the term chromosome, which have the affinity to basic dyes. Hence he just coined that the chromosome word. And for this this chromosome can be easily counted in the different cell division of mitotic metaphase. And we know very well that each human normally has 23 pairs of chromosomes, that is 46 in number. And in each and every living organism there is a diploid level 2n number is there, as reflect the chromosome number, somatic chromosome number you can say. So now we come to the chromosomal aberration. It means aberration means the abnormalities, the anomalies, disorders observed in the chromosomal DNA. It may be caused by the different type of mutations, maybe a spontaneous mutation or induced mutations. However, this abnormalities is very meager in their occurrence. Then that it occurs, for example, it occurs 5 to 6% in 1,000 persons of this human being. So its occurrence, its incidence is very meager. However, this chromosomal aberration play a very effective roles in their different origin, the evolution, and some other speciation and some other variations. So the children with chromosomal abilities have mental or physical defects by birth. This type of if any progeny or any individuals carrying this chromosomal aberrations, they show their retardedness right from the birth. So some scientists like Jerome Marthe and Raymond Turpin discovered the presence of an extra chromosome in Down syndrome patient in 1959. The next one, the Ford, Jacobs and Strong first observed 6 chromosome abnormalities in the patient having sexual development disorder. So when we consider the different types of chromosomal aberration, this chromosomal aberration may be of a structural and of numerical chromosomal aberrations. A structural chromosomal aberration means the chromosome, the abnormalities of chromosome in their morphology, in the genetic sequence, in the genetic you can say that kind. So this type of aberration is taken place. But in contrast to this numeric structural chromosome, numerical changes plays a different roles in this in the individual, in the living body, that it may be of monosomic, it may be of trisomic, where deletion or duplication of addition of one pair of chromosomes takes place, and the nullisomic, where the one pair of chromosome is completely lost. So these are some of the numerical chromosomal aberrations. But in this class, in this lecture, we will concentrate on the structural chromosomal aberration, that means for the morphological differences, morphological modification, deviations from the normal chromosomes. This structural chromosome may be intrachromosomal and interchromosomal type. These are two different types of structural chromosomal aberrations. We can see here the chromosomal structural abnormalities and different form, different phenomena, phenomena. So structural chromosomal aberration cause the, as I told, that causes the abnormalities in structure, in the morphology, in the sequence of gene is present on the chromosomes. They alter the sequence or the number even, or the kind of the gene present in the particular chromosomes. Here one thing must be noted, that here the chromosome number is not modified in any way. So as we heard that the structural aberration is maybe of intrachromosomal and interchromosomal. So in the case of intrachromosomal aberration, the modification of chromosome confined on a single chromosome, and then this modification may be due to the different physiology, different phenomena like deletion, duplication and inversions. While as in case of interchromosomal aberration, this modification, the structural changes takes place on the non-homologous chromosomes, and the resulting fragment are interchanged by both the non-homologous chromosomes. And it may be this type of chromosomal aberration, structural chromosomal aberration in interchromosomal system takes place by the phenomena of translocation and insertion. So we will discuss each and every phenomena which are responsible for making the morphological variation, morphological modification in the chromosome. So, and for that, the first one is the deletions. Deletion, as it is literally means that the deletion of anything, the things, deletion, it means just removal of any things is called the deletions. So similarly here in case of chromosomal aberration, when a part of a segment of chromosomes is removed from the normal counterpart, it is called, the phenomena is called the deletion. The deletion, it is very much important. It plays a very vital role in particularly in human being, and it is very much deleterious. It may occur due to the losses from translocation, and sometimes crosses over within an inversion, and sometimes unequal crossing over takes place that resulted this type of phenomena and this deletions. Here in this case, the breaking of chromosome is only takes place, the reunion is not there. Rejoining of the segment part is not taken place in this deletion. Once it is break, and then it is lost. You can see here the photographs of deletion, where in the normal chromosomes 2 breaks takes place, and that the segment part of chromosome, segment of this chromosome is deleted, and you can see here the ultimately the resulted chromosome is shorter in the length as in compared to that of the normal one. So types of deletion, there are different types of deletions. It depends upon the removal of chromosomal segment from the normal chromosome, that is maybe the terminal deletions. In this case, the terminal segment losses from the normal chromosomes, and here in this case a single break is required. A single break in the chromosome takes place, and the segment of that chromosome is lost. We can see here the origin of terminal deletions in these photographs, that the break takes place in between the A and B genes, and then A genes is lost from the normal chromosome, so the resulted chromosomes is only containing the B, C, D, E and F genes, whereas the segments carrying the A gene is lost. Second type of deletions is the intercalary deletion. In this type of deletions, two breaks is taken place on the normal chromosomes, and then internal part of chromosome is missing. So here you can see the origin of intercalary deletion means the break takes place in between the different genes. So here you can see the two breaks at one at B and D portion, and another one is the C and E portions takes place, and they make a loop. And then ultimately that loops is lost, and again this other one, the chromosome, the resulted chromosome to get together, and then you can see the resulted deleted chromosome having only A, B, E and F genes, but the chromosome segment which carries the C and D genes here is lost. So some of the genetic effects of the deletion, as we already told, that it is very much vital, particularly for the human beings. It expresses the different physiology in the human being. So it generally produce the genetic and morphological physiological consequences. The morphological and physiological consequence takes place in this deletion, due to this deletion. If that deletion or deficiency is larger type, then it produces sometimes the dominant lethal effects. Lethal effects means the individuals is not able to survive. They are bound to die. So this type of deletion sometimes, if present in larger size, then in that case it may result the lethal effects. Next, if the loss of genes are critical, sometimes some of genes are very much essential for their viability, then in that case the non-functional somatic cells are resulted. In human being, this genetic deletion mutation is caused by the mutation, and this some of the characteristic, the disorders, you can say the syndromes is taken place, like Cray-Duchet syndrome, while the deletion of entire chromosome of 5 takes place. So another example is the in Drosophila, when deletion of 3C7 band of X chromosome takes place, then it gives rise to a notch wing Drosophila. Next one is the duplications. As it is literally understood that literally means that duplication means addition. It means the doubling, the repetition of the chromosome here. In this there is doubling or repetition of chromosome segment during chromosomal duplications. In this phenomena, while takes place, there is duplication or the doubling of the chromosome segment takes place. So the extra part which is being doubled or which is being added to the normal one is called as the repeat. REPEAT. Repeat means the same segment is repeated on the normal chromosomes. So first, this phenomena of duplication was detected by the Bridges in the year 1919, while they study the X chromosome of Drosophila. It involves in this physiology the chromosome breakage and reunion of the chromosomal segment with its homologous chromosomes takes place. If we compare the deletion and the duplication, then we can clearly differentiate that in case of deletions, only the breakage is taken place. There is no question of rejoining, but here in case of duplications, there is both the process of both the steps of chromosomal breakage and the rejoining takes place, but it always be, it should be, and the rejoining should be reunion should be with its homologous chromosomes. Sometimes unequal crossing over may takes place, and that resulted the duplication phenomena. So for example, in case of Drosophila, the duplication, the doubling of 16A of X chromosome resulted the bar eye color. So if we talk about the types of what are the different types of duplication, that the addition, the doubling is taken place on this normal chromosome in different way. That first one is the tandem duplication. In this type of phenomena, this type of process, the doubling of addition of chromosomal segment located just after the normal segments. So here the gene sequence is same as is the original one, but in case of reverse duplications, the extra chromosomal segment which are added to the normal chromosome is just next to the parental segment, but the additional segment, the genes or the sequence of gene present on that the segment is additional segment is inverted, is just reverse. Next one is the displaced duplication. In this type of duplication, the additional segment, the added segments is placed somehow distantly to the original ones. It means that it is may some away from the original parent segments. And last one is the translocation duplications. In this duplication process, the additional segment is located on the non-homologous chromosomes. So we can see here the normal chromosome that containing the 1, 2, 3, 4, 5, 6 genes, and here in case of the process of tandem duplications, we can see here the breakage of 2 and 3 takes place, and then that segment is repeated just the parental segments and in the same sequence 2 and 3 you can see here. But just in contrast to this, we can see in the case of reverse duplication that 2 and 3 segments containing the segment which containing the 2 and 3 number of genes is just placed near to the next to the original segment, but the sequence of that are just reverse, just you can say that just reversed segments. The placement of these genes are just reverse. So you can see this. But another one is the non-tandem duplications. We can see here the segment of containing the 2-3 genes is cut and then just duplicated duplicated at the position where at the 6 and 7, in between the 6 and 7 genes, not near the original parental segments. So these are different, you can clearly understand the different types of process, different types of duplication in the chromosomal aberration. So some of the genetic effects of duplication is it is less deleterious as compared to that of the deletion. We are already told that the deletion is very much deleterious, vital. But as compared to this genetic duplication is little bit less deleterious to that of the to individuals. So in this case, duplicate, complementary and polygenes are originated. The genes may be the complementary genes, maybe the duplicate genes or the polygenes are originated to this process of duplications. Origin of sometimes new genes are also takes place that has the distinct functions that may be due to the increased DNA content per cell. In each cells there is a fixed amount of DNA content, but if a particular chromosome has this process, this type of duplication, then the new genes may have the distinct functions, and that is resulted due to the duplications. If the larger duplications takes place, then that affect the regulation of gene activity, and thereby that causes the phenotypic alterations, which in turns promotes the speciation, the process of evolution, the process of speciations, the creation of new individuals to that of the older ones, existing one. We can see here the bar eye phenotype in Drosophila as the result of this duplication. Next is the inversion. This one is the last one, that the intrachromosomal aberrations. Then in this case it refers to the reversal or just reverse of the linear order of a chromosomal segment and its sequence both. This inverted segment, whatever reversal takes place, and that rotated by 180-degree angle, and so it make a loop. This inversion, the process of inversion was first detected by Stutter, Brandt and Plunkett in the year 1926, and this type of inversion can be readily detected while we prepare the linkage map, and but in absence of crossing overs. Inversion loop can be observed. This what I told, that the loop is formed, and that loop is observed easily during the Pachytene stage of cell division. If we talk the types of inversions, there are two types of inversions. First one is the paracentric inversions, and another one is the pericentric. As it is literally clear that paracentric, it means the centromere is located outside the inverted segments. Those inverted segments doesn't have the centromeres. It do not alter the morphology of affected chromosomes. But in case of pericentric inversion, the centromere is located within the inverted segment and within the loop, so it may alter the centromeric positions. This is the two major difference between this type, the paracentric and pericentric inversion, that the place of centromere and the alteration of the centromeric positions. This is very much important. So we again repeat, the paracentric means the centromere is located outside the inverted segments. Inverted segments, I told earlier that it makes a loop, and that loop doesn't contain this centromeres. But in case of pericentric, that loop has this centromere, and the centromeric position is altered in case of pericentric inversion. We can see here the paracentric and photographs of paracentric and pericentric inversion. Here we can see the normal chromosome, and breaks in chromosome takes place, and this inversions in this just rotate. The green part here is at bottom level, but when it rotates at 180-degree angles, you can see here the reinserted piece of DNA is just inverted, just reverse to that of the original one. So green part of this the chromosome is at upper portions. You can see here the position of the place of centromere remains same, because the inverted part doesn't contain this centromere. The centromere is placed outside of the inverted segments of this DNA. In contrast, here you can see that in case of pericentric inversion, the breaks takes place, and that is in between, that is at the place where the centromere is located. This inversion takes place, and here the similarly the just inverted, the DNA segments is inverted, and you can see finally the resulted chromosome is the reinserted piece of DNA with centromere. Here you can see this inverted segment of DNA. Effects of inversion that helps to retain the original gene combinations by reducing the crossing over. It enhances the position effect. Position effect is very much important in case of agriculture science, and thereby it produces phenotypic change. We can see the different phenotype in the crops, in different crops. It causes chromosomal polymorphism within a population that leads to the karyotype evolution, the evolution based on the karyotype. It produces the balanced gene complexes through reduction of crossing over, which in turn confers the greater biological fitness and adaptability and increases the evolutionary potentiality. The adaptability, the fitness in the different different climatic condition takes place by this producing balanced gene complexes, and this sometimes the male sterility also takes place due to this inversion, that is partially, not totally, it is partially male sterility takes place. Formation of recessive mutation is the effects of this type of phenomena. Now next one is the translocation. Translocation, it means literally we can see that the movement, the just transmission of a segment of a chromosome to the non-homologous chromosomes. Here two pieces of different chromosome are interchanged, and one portion of one chromosome is transferred to the another chromosomes. Formation of cross-shaped configuration between two non-homologous chromosomes takes place, and here you can see the ring of 4 chromosome due to pairing of homologous chromosome as metaphase 1. Here the different types of translocation is the first one is the simple translocation. In this, the terminal segment of a chromosome is integrated at one end of the homologous or non-homologous chromosomes. Here we must understand the translocation, this process involves the non-homologous chromosome. Here single breaks takes place, and this simple translocation is reported by Painter and Muller in the year 1929 while studying with the Drosophila. The another one is the shift translocation. It is an intercalary segment. Here the intercalary segment is integrated in an intercalary position of non-homologous chromosomes. It means the segment is whatever breaks takes place, that is in between the different genes, and not at the terminal end, but it is in the intercalary places, and that is breaks, and then that segment is integrated into the intercalary position, not at the terminal position. Always we remember that neither the break takes place at terminal end, and nor their integration is also takes place at the terminal ends. Here the intercalary, the segment which is just going to be translocated is breaks from the intercalary place, and then that segment is integrated at the intercalary places of the non-homologous chromosomes. So it requires 2 breaks in one chromosomal, while only 1 break in translocated and non-homologous chromosomes. We talk about the breakage. Breakage takes place at 2 different places because it is in between the intercalary places. The break takes place at intercalary, not the terminal. If it happens at the terminal end, so only one break is required. But here, in case of this shift translocation, the segment which are going to break requires two breaks, one and the another one. So this segment is then translocated to the non-homologous chromosomes. Then the only one break is required at the non-homologous chromosome. So in total, there are 3 breaks takes place in this process, and it is known in Drosophila and Neurospora. Another one is the reciprocal translocation, and it is very much common and widely studied. Here in this case, a segment from a chromosome translocated into a non-chromosome from which a segment is reciprocally transferred. So here only one break, single break is required. So you can see here the photographical representation of this different type of shift and reciprocal translocations. Here in this case, we can see the B and C segments is just translocated to the non-homologous chromosome of this of the non-reciprocal translocation. Here in this shift type, the B and C chromosome segment is translocated to the B and C, the non-homologous chromosome of MNOPQR. But here in case of reciprocal translocations, you can see here that both the segments of the chromosome, the original one and the non-homologous one, is interchanged. The segment part is interchanged, though this the length of segment varied, but they interchange at the in the approximately in the same end and same places. Here you can see the AB segments of the original chromosome is translocated to the another non-homologous chromosomes, and the chromosomes of non-homologous segment, the segment of the non-homologous chromosome is just translocated to the original, the ABCDEFGH chromosome which carry this gene. So you can see here also the photographs of different types of translocation. Some of the effects of this translocation that alter the chromosomal morphology. It promotes the variation, it creates variation, which is responsible for the evolution, the speciation. Sometimes the position effect, which is very much important, the position effect is also due to the translocation process of chromosomal aberrations. Damage to DNA may sometimes resulted the formation of recessive lethals. It causes the disease like cancer in human being, and it leads to the impaired fertility sometimes. Complication in synopsis and segregation also takes place due to the effects of this translocation process. The last one is the insertion. It is a process, it is an event in which the piece of chromosome is removed and inserted in the different or another chromosomes. In this case, inserted segments of DNA can also move from chromosome to chromosome, from one chromosome to the other chromosomes. It free to move from different chromosomes. It resulted in loss of genetic material from one chromosome. Here it is very much important. So you can see here the photographs of this insertion. Before insertion, we can see here the normal chromosomes of 4 and chromosome 20, and just the part of this inserted segment of chromosome 4 is inserted in the chromosome 20, so the genetic makeup will change. Another one morphological differences or morphological changes in chromosomes is the type of ring chromosome. Ring chromosomes. It is a structurally abnormal chromosome in which the end of each chromosome arm has been lost, and that broken arms is reunited to form a ring. We can see here in the photographs the breakage takes place at two terminal end of this chromosome, and that terminal end, that segment which has been terminated, which has been deleted, we can say breaks deleted from the original one, that just reunite and form a ring, and then this is the all other one is fused. Now, another one is the morphological changes of chromosome is the isochromosome. The product is the isochromosome. In this case, it is unbalanced structural abnormalities in which the arms of chromosome are mirror image of each other. The chromosome consists of two copies of either the long arm or the short arm, because isochromosome formation is equivalent to a simultaneous duplication and deletion of the genetic material. When duplication and deletion takes place simultaneously, then in that case this type of chromosome is resulted. Another one is the derivative chromosome. It is structurally rearranged chromosome that generated either by rearrangement involving two or more chromosome or by multiple aberration within a single chromosome. That is, an inversion and a deletion of the same chromosome takes place simultaneously, or the deletion in both arms of a single chromosome when takes place, then the resulted chromosome is called as the derivative chromosome. Some of the uses we should know that about, or we must know about the structural aberrations. Then how the significance of this structural aberration in the agriculture science or in the life science takes place. So the study of chromosome, it helps in the study of chromosomal pairing and their behavior during the cell division, how the chromosome behave during the different stages of the cell division. Next one is the locating the genes, where the particular genes are located on a particular particular chromosomes. For this structural aberrations is helpful. Sometimes it produces also the new genes like duplicate genes and some other genes, polygenes is produced with this structural aberrations. It is useful in studying the position effect of the genes, is very much important, and it plays a greater role in the process of evolution. Sometimes desirable characters can be detected by the inversions. Translocation may also be helpful in chromosomal mapping. The process of paracentric inversion is used to produce the acentric fragment intact. It is also this significance of this chromosomal aberrations. So the role of a structural chromosomal aberration in plant breeding, when we talk about this, it is very much useful in identifying a particular chromosomes, and that is all that utilization of the vigor is what is the estimation? We can say that first we estimate the potentiality of the vigor, and then this is utilized in case of duplications. Sometimes it is also useful in genome analysis, and it also be useful for the transfer of desirable characters through translocation. There are different process of this chromosomal aberration, and lastly the evolutionary significance is much more with this chromosomal aberration. Some of the structural aberration chromosomal structural aberration on the human being, then there are different syndromes takes place by deletion or deletion of different chromosome. For example, while microdeletion of chromosome 5 in human takes place, then it produces the cry-do-chet syndrome, and other, in contrast to this, when 22 number of chromosomes is deleted, then it produces again another type of syndrome, that Di-George type of syndrome. And if the 15 chromosome number is deleted from the human being chromosome, then it produces the Engelmann syndrome. It is very much dangerous, deleterious. And some of the reference we can see here for this lecture. Thank you very much.